Assalamualaikum warahmatullahi wabarakatuh. Just a quick testing on our audio. Thanks everyone for attending the session. Just for confirmation, can you listen to my voice clearly, or is it echoing?
Yeah, very clear.
Prof. Shariza, can you listen?
Yeah, I can hear you.
Prof. Muhammad Lokman?
Yes, very clear, inshallah.
Dr. Nurul Ain?
Yes, it's clear over here.
Alhamdulillah. If that's the case, then can we start the session now?
Inshallah.
Yes, inshallah.
Inshallah. Bismillahirrahmanirrahim. Alhamdulillahi Rabbil Alameen. Wassalatu wassalamu ala ashrafil anbiya wal mursaleen wa ala alihi wa sahbihi ajma'in. A very good morning to our honorable speakers for today's webinar. I wish to welcome all of you to the stem cell research seminar. So this is the first series of our stem cells webinar for this year. And before we go further, let me introduce you about what is the aim of this seminar. So we aim to expose all the IIUM community and also some learning institutions as well as the industries regarding the importance of stem cell research, particularly in Malaysia. So this is also to create some awareness among the postgrad students as well in carrying out their research work. So we hope that all the students can use this seminar to gain some benefits or networking to find their suitable supervisors or their suitable examiners for works related to the stem cell research. Just for a housekeeping announcement, we will advertise this webinar on the Facebook Live in the IIUM Molecular and Cellular Biology Research Cluster Facebook page. All the registered attendants will be provided with the electronic certificates, and we hope that towards the end of the session, you can provide the feedback form to us. And throughout the session, we will give the chance for the people to ask any questions related to the topic highlighted by our speakers. And please mute your microphones so that it will not interfere with the speakers while they are speaking. Without further ado, let me introduce our speakers for today's webinar. Our first speaker is Associate Professor NORSHARIZA binti NORDIN. She received BSc from California State University, Sacramento, United States of America. She did her MSc in Universiti Malaya, and after that she continued with her PhD studies in Edinburgh, United Kingdom. She is currently the head of medical unit in the Department of Biomedical Science, Faculty of Medicine and Health Science, Universiti Putra, Malaysia. And she is holding a position in Genetic Society of Malaysia as the Vice President. She is also the founding editor-in-chief for Neuroscience Research Notes. As for today, Prof. NORSHARIZA will explain and also will discuss with us what are the view and also the perspective of stem cell research, particularly in Malaysia. She will be talking about her title, which is Pluripotent Stem Cell. What is it for us in research? Be mindful that all panelists have 20 minutes for their presentation, plus 5 minutes for Q&A questions. Without further ado, I will begin with the first speaker, which is Associate Professor Dr. NORSHARIZA binti NORDIN with her talk. Please welcome.
Bismillah. Thank you very much, Dr. Tengku Faris, for a very nice introduction. Let me share my slide. Can you see my slide? Hello?
Yes, we can see.
Can you see my cursor?
Yes, it's moving.
Okay, all right, good. Thank you very much. All right, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And a very good morning to everyone. Thank you very much, Dr. Tengku Faris, for a very nice introduction. And then I also wish to thank Dr. Tengku Faris and also the organizing committee members for giving me a chance to share a little knowledge of mine and also to share some findings of my private topics that is on pluripotent stem cells, and which I will be sharing also about on the perspective of how to move forward. Yeah. All right. So before I start, so what I'm going to before I begin, yeah. So I would like to share that what I'm going to discuss about during this talk. So I will be talking about what are stem cells. Yeah, for those who are not so familiar with stem cells, I will tell about what are stem cells and how to get them and what we can and we cannot do with them. Yeah, and then also share with a bit of our findings, yeah, my team's findings, and then how to move forward with pluripotent stem cells. All right. So for those who are not familiar, so during development, yeah, so we have the existence of, the stem cells in our body. Yeah, upon conception, we have a population of when upon fertilization, yeah, upon conception, we get a population when the development reach the blastocyst stage, yeah, the early blastocyst, there will be a mass of cells that's called as inner cell mass, where from this mass you get to develop into various cells in our body. So in embryo, so this inner cell mass contains a population of cells that is known to be pluripotent stem cells. So in embryos, these stem cells function more to generate new organs and tissues in our body. Yeah. So at the end of the day, you get the whole organs and tissues in our body. Yeah, we have all the internal organs in our body. And then in adulthood, after we are born, when we are babies, and also during adulthood, we also have stem cells all over our body. So the stem cells now is more function to replace the damaged or lost cells, yeah, because of the natural cause we have cell turnover in our body. So the stem cells during adulthood, or adult stem cell, we call it, is meant for act as a spare part, yeah. And they're very localized throughout our body. So in other words to say, throughout our body, we have multiple types of systems in our body. So each system contains its own sets of stem cells, yeah, as an adult. All right, so what are these stem cells? So those are in vivo. What about in vitro? Can we isolate and grow them in vitro? Yes, we can. Yeah, so that's why we are here today, to talk about the values or the properties of these stem cells in vitro scenario. Yeah. So basically, what are stem cells? So stem cells are undifferentiated cells. So that means and also they can make more of themselves. So that is why making the stem cells such a valuable cells because they can provide a continuous supply. Yeah, so it's very good for clinical application. So then we have, so that means they are undifferentiated, so unspecialized cells, so which they can make more of themselves, they can replicate. And the other important thing is that they can make, they can be differentiated, or they can differentiate into various types of cells in our body. Yeah, not only can differentiate into specialized cells, but they can also differentiate into functional cells. Yeah, functional cells. For example, under appropriate physiological condition, you can make stem cell to become cardiac cells, but not just a cardiac cell, they can also become a beating cardiac cell. Yeah, so these are called differentiated cells. So these are the two main characteristics of all types of stem cells. Yeah, moving along, during development, you can find different types of stem cells with different degree of stemness. So we refer that as potency. So as you can see, the pluripotent stem cells, yeah, you can get it from the early stage zygote, yeah, until cell stage. You can get pluripotent stem cells. So by all means, pluripotent means you can from pluripotent you can get the whole organism. Yeah. Then we have pluripotent as the blastos, when the zygote reach the blastocyst stage. Now they become the embryo. So I mean the not yet the embryo, still the zygote, but at the early stage, the blastocyst stage, you can get the inner cell mass, as I mentioned earlier. So you can isolate, you can get embryonic stem cells, and also you can get primordial germ cells. So we can get from primordial germ cell, you can get embryonic germ cells. So these are categorized as pluripotent stem cells. So the pluripotent stem cells can give rise to all the three primary germ layers, but not the extra embryonic tissues such as placenta. Yeah? Some they can, but not the placenta. So because of that, pluripotent stem cell cannot become the whole organism. Yeah, they cannot become the whole organism. So they can differentiate into all the cells in our body, the derivatives of the three primary germ layers. Yeah, and then this specific germ layer, they will then develop, yeah, in vivo into a specialized into their lineage. Yeah, so each lineage then have their own stem cell. So that stem cell is multipotent. We call that multipotent because they have a limited, they can differentiate into a limited number of cells, yeah, from their own germ cells. And then we also have unipotent. Yeah, so today I'm going to talk about pluripotent stem cells. Yeah. So let me introduce to you the FAMES and also those who are responsible in making what we know now about the technologies that we can use to with pluripotent stem cells. Yeah. So extensive studies have been going on for decades, yeah, which have provided us with the current knowledge and technologies on stem cells, particularly on pluripotent stem cells. So since 1963, yeah, an idea that nuclear cloning from somatic cells is possible came from the work by Sir John Gurdon, yeah. So which then lead to the and then in 1981, yeah. So the work by Sir Gurdon implies that we can actually reprogram somatic cells. Yeah, okay. And then from his work from cloning using in frogs. Yeah, okay. And then in 1981, the early 90s, a group of Martin Evans and Merty Kaufman, they managed to isolate the first mouse embryonic stem cells. Yeah, in 1981, and then which then inspire James Thomson to try to isolate this population of this pluripotent stem cell from human. Yeah, so back late 90s, only then we managed to have human embryonic stem cells being isolated from human. Yeah, and then we have a work from Ian Wilmot and colleagues from Roslin Institute, yeah, of the University of Edinburgh, that lead to the birth of doll leadership. Yeah, doll leadership. This work is very important as it demonstrates the ability of adult cells or somatic cells to be transformed into embryonic. So as a whole become a whole whole organism. Yeah. So in that sense, the work implies that nuclear reprogramming of adult cell is possible, which can lead to the discovery of induced pluripotent stem cells by Yamanaka from mouse fibroblasts in 2006, and then from human, iPSC from human by Takahashi. Yeah. And then ever since then, more advanced reprogramming technologies were developed, until we get these technologies for generating pluripotent stem cell using various technologies. Among all, this is the conventional method where we isolate the inner cell mass from, we isolate the stem cell, the pluripotent stem cell, so from the inner cell mass of an embryo, early blastocyst stage embryo. So this is what we call embryonic stem cells now. Yeah, and then the work by the creation of Dolly also has managed to lead to the creation of embryonic stem cells from somatic cells. Yeah, so that is a technique called somatic cell nuclear transfer technique. Yeah, so we get somatic cells and then being fused into the enucleated egg. So we have these donor cells. Then we take the DNA of the donor cells, and then we have also to get an ovum, a donor ovum. So we take out the nucleus of the donor ovum, and then we fuse them together, and then we give them a quick electric pulse to stimulate cell division. The cell now can divide because it has complete chromosome, because the complete chromosome comes from the adult DNA, the somatic cell, the donor, and then you can create the embryos. So we stimulate the fusion of this donor nucleus and also the egg, the ovum, and then we stimulate using electrical pulse. They divide, and then later on they become the embryo, and then we can then take the embryo and then proceed and treat it as the embryonic stem cells. So this technique is being popularized due to the inability of the embryonic stem cells for autologous transplantation. So using somatic cell nuclear transfer technique, because the donor cells, the DNA is actually similar to so you can create the embryonic stem cell that is genetically identical to the donor. So for example, a patient. So now you get the patients donate the cells, which contain the DNA. So you can reprogram DNA and make clones of the patient. So now you can get the embryonic stem cells here, and then they can differentiate into whatever cell that is needed by the patient. So this is a technique called somatic cell nuclear transfer technique, which also can generate embryonic stem cells. And then a technique also you can generate pluripotent stem cell also from single blastomere isolation. So this is a technique now being practiced for PGD, pre-implantation genetic diagnosis. So you can isolate one of the blastomere, and then now we know that we can grow this single blastomere to complete embryo, and then we can take the embryo and to establish to isolate the pluripotent stem cells. And then lastly, the work by Yamanaka, and the rest. Also, now we have technique has improved in reprogramming of the somatic cell to generate what we call induced pluripotent stem cells. So this is reprogramming comes from the differentiated cells, and then there's various reprogramming method that can be done using either genetically modification method, or you can do small molecules. So there are a lot of factors now, a lot of methods you can use to reprogram somatic cells or differentiated cells or mature cells, into induced pluripotent stem cells. All right. So along the development, as we mentioned, so you can isolate embryonic stem cells, but scientists also have discovered there are different types of pluripotent stem cell or pluripotent population that you can harvest throughout development. And then they have classified embryonic stem cells are pluripotent stem cells that you establish pre-implantation. Yeah, so that means pre-implantation. So this is from pre-implantation embryo. And then you also can get a population of pluripotent stem cells from post-implantation embryo, which is called epiblast. So the isolation of pluripotent stem cell from epiblast is called epiblast stem cells. And then you can also isolate pluripotent stem cells from late or embryos or very early fetus. Yeah, so that is from the primordial germ cells, which is called embryonic germ cells, also pluripotent. Yeah, and then as development proceeds, you get multipotent and unipotent, as I mentioned earlier, and then you can get you can get our bodies to differentiate, to have different kind of lineages in our body as an adult cell or differentiated cells or mature cells. And then now you can reprogram the cells to become induced pluripotent stem cells. Yeah. And some group also, they have discovered that a descendant of epiblast stem cells, yeah, that is passed through into adulthood, into adult tissues, particularly in the bone marrow, where they isolate, where they manage to isolate cells, yeah. For example, V cells, stem cells. V cell stands for very small embryonic-like stem cells. And also some claim MAPC, yeah, sorry, spelling error, MAPC, which is the progenitor cell, a multipotent adult progenitor cells, yeah, MAPC, not MAPS, sorry, my spelling error. And then Miami cells, which is the marrow-isolated adult multilineage inducible cells. This also, some has coined them as or categorized them as pluripotent stem cells. Yeah? These are from bone marrow. They are believed to be descendant of epiblast stem cells, right? And then scientists have done a lot of studies and then comparing the genetic profiling of the in vivo blastocysts and also in vivo epiblast, and then they have actually categorized and they have actually concluded and come up that the pluripotent stem cells can be divided further into two categories. Yeah, one is called as naive pluripotent stem cells and also prime pluripotent stem cells. So what are the difference between these two are that based on mouse studies, yeah, based on mouse studies, they have concluded that the naive pluripotent stem cells actually from the pre-implantation ICM or the pre-implantation epiblast before it being implanted into the uterus. And then these cells, when injected into the pre-implantation blastocyst, they can contribute to chimera, sorry, spelling it again. Chimera, they contribute to germline transmission and they can give rise to, again, to all the three embryonic germ layers, and they can form teratoma and also they are hypomethylated. And then the prime cells, they actually derive from post-implantation epiblast. That means after the embryo has been implanted into the uterus. So these are post-implantation epiblast. They can contribute to chimera when injected into post-implantation epiblast. So they're not from the early stage. You cannot get, they cannot grow when you inject it into the blastocyst stage. Sorry, spelling error. And then they give rise to all the primary germ cells still, yeah, that's why they're pluripotent. And then they can form teratoma because they can give rise to the three primary germ layer cells and they are hypermethylated. So these are the difference between naive and prime cells, yeah. If we proceed with the transcription factors and their signaling pathway that are involved in pluripotency, you can divide it, the networking into three parts, three categories. One is core pluripotency markers, and you also have extended regulatory markers or factors, and also you have signal transaction pathways. Yeah. So you have OCT4, which is very important. OCT4 is very important. So these three are core pluripotency. So if you publish a paper claiming that you have established an embryonic stem cells or any pluripotent stem cells, the journal wants to see you show them the expression of OCT4, SOX2, and NANOC. Yeah, these three or at least two. So one is OCT4. OCT4 is very important because it is transcription factors that lead to the expression of target genes that maintain pluripotency, and also SOX2 that regulate the function of this transcription factor is to regulate embryonic development, and also it prevents AS cell differentiation. So it maintains the cells as an undifferentiated cells. Yeah? And then you also have NANOC. These transcription factors work hand in hand with OCT4 and SOX2, yeah, to improve cell proliferation, survival, and maintenance. Yeah, but sometimes you don't need to have this directly. You also can you can get NANOC in the system by having KLF4, because KLF4 is cofactor for OCT4. Yeah, it also represses P53. Yeah, we all know that P53 actually negatively regulate NANOC. So when it represses P53, so it means it regulates NANOC. Yeah, and then CMYC. CMYC is an effector of STAT3. STAT3 is important to maintain pluripotency. So if you can see, and then also the signal transduction pathway, which is Wnt, Notch, TGF beta, Hedgehog, Leaf, and fibroblast protectors, and also insulin. These are the additional signaling transduction pathway that is also needed or helping in maintaining the pluripotency. If you look at the transcription factors, you can see that the KLF4 and actually CMYC actually can replace NANOC. Yeah, so that's why if you look at the reprogramming factors proposed or used by Yamanaka, so we know this OSKM as Yamanaka's reprogramming factors, you can see that they use OCT4, SOX2, KLF4, and CMYC. Yeah, because these four will then compensate the need of using NANOC in the system. Yeah. All right, let's look at how we can get embryonic stem cells. So embryonic stem cell, in particular, if human, you can get the source if from the embryo, a very early stage, yeah, either ICM or epiblast. Yeah, so you can get from 5 to 7 day old embryos from human. If it's from mouse is actually about 3.5 to 4.5 day old embryo. So you get, they actually, you can see this is very, very, very young, very, very early stage embryo. So you can imagine this is not, you cannot get this embryo from a normal conception, normal way of conception. So it has to come from IVF. Yeah. So all human derivation or establishment of human pluripotent embryonic stem cells, yeah, they actually come from assessed IVF embryo. Yeah. So you get upon in vitro fertilization, yeah, IVF, you get the embryo, the zygote, and then you get the blastocyst stage, yeah. So the way you do it, you get this, once it reaches the blastocyst stage, you know there is the existence of the inner cell mass in the blastocyst stage, yeah. The inner cell mass which will give rise to the 3 primary germ layers. Now we isolate this inner cell mass, yeah, when we culture them. So they become cultured pluripotent stem cells. Yeah. So there are various methods of how you can do or derive these inner cell or these embryonic stem cells. Yeah. But I don't have enough time to tell you all that here. Yeah. All right. Okay. So the controversial issue here is that the derivation or establishment of human embryonic stem cells will need a sacrifice of an embryo, because once you isolate out the inner cell mass, then you will then destroy the embryo. You will not proceed to organism proper, yeah, because you already took out the inner cell mass. Even then, some people claim you can get from the blastomere, you take only one blastomere, right? But there is still an issue for human. You still an issue to do that, yeah. Still controversial. So that's why it's quite controversial. And then they can differentiate. So this is how you get the morphology of human embryonic stem cells, yeah, in the dish. So you get, it has to be grown on the feeder layer, yeah, and then this is how the morphology of embryonic stem cells look like in a dish. Right, so they can actually differentiate into all the cell of the primary germ layer, so about 222 cell derivatives. So they can, but they cannot form one of the extra embryonic tissues, which is the placenta. Okay, the characteristics of embryonic stem cells that they can form embryonic bodies, they can form teratoma when injected directly into the host, into the body. So they allow for allogeneic transplantation, but they don't allow for, because if you can see, if you direct in vitro fertilization, you take from mother and father, from sperm and ovum, so it's not identical, so you cannot get these cells to be transplanted into an genetically identical person. So it's called allogenic transplantation. So it doesn't allow, or it doesn't permit for autologous transplantation. But now the somatic cell nuclear transfer technique will allow you to do that. Remember the somatic cell nuclear transfer technique that I was mentioning earlier, right? And then induced pluripotent stem cells, thanks to these people. So now we know that we can actually get somatic cells to be reprogrammed into induced pluripotent stem cells. So the reprogramming, now starting from fibroblasts, and now we get various somatic cell source that people have reprogrammed them into induced pluripotent stem cells. So there are different methods. One, you can either use genetic manipulation technique to reprogram the cells, or you can use chemical reprogramming to reprogram the cells. So there are various types of reprogramming techniques, but bear in mind, different source of somatic cells, different reprogramming technique will lead to different efficiency of the induced pluripotent stem cells. So the characteristic and differentiation potential are similar to embryonic stem cells. So because the source is from the somatic cells, so it does not involve invasive technique. So you can get any cell of a human being. So in particular, if you want to make it like patient specific or disease specific, you can get it from a particular patient suffering for a particular disease. So you can reprogram it to make it patient specific. So you can use it for disease modeling or drug discovery or even personalized medicine. And then you can also correct the patient's induced pluripotent stem cells. We do gene therapy, gene correction, and then you can transplant back to the patient. So now this transplantation we call this autologous. So it allows for autologous transplantation as well as allogenic transplantation if the source did not come from the same patient, the same person. And you can also generate the induced pluripotent stem cell from normal patients and from normal individual. So you get normal iPSC, and then you can use it for drug development, or you can also use it for biobanking. So a lot of countries now has come up with the idea of pooling iPSC, and then they have this biobanking of induced pluripotent stem cell lines where they get it from different people, and then they screen for the HLA subtypes. Right. So this is how the induced pluripotent stem cell look like. They look similar to the embryonic stem cell. They look similar, but they're not 100% the same. Bear that in mind. Okay, so along the way, we know that we can get different pluripotent stem cells from inner cell mass, or either from epiblast, or we can reprogram it. But there are different categories. So scientists have categorized mouse embryonic stem cells generated from inner cell mass together with mouse induced pluripotent stem cells to be categorized as naive pluripotent stem cells, whereas those who are mouse epiblast or from epiblast, post implantation mouse epiblast stem cells, they have similar category as human embryonic stem cells and human induced pluripotent stem cells. So these are categorized as primed pluripotent stem cells. So bear in mind, the naive is actually mouse embryonic stem cell and mouse iPSC, prime mouse iPSC, as well as human iPSC and human embryonic stem cells. So these are primed category. All right. So the importance of prime and naive is that they have slightly different different characteristics and also different properties. So people have tried to understand what are the global, what we call the hallmarks of naive and primed. So one of it is that they are unique. They have, each of them has unique set of transcription factors. Yeah. Even though both will express the core pluripotency markers similar, OCT4, SOX2, and NOx still maintain the same, but they have sub-specific markers. They have a unique sub-specific transcription factors that they express. And then they also have a different global methylation status. Yeah, so they have for naive, they are hypomethylated, as I mentioned earlier, whereas for prime, they are hypermethylated globally, yeah, global status. And then in terms of the X chromosome status, the naive also, they have two, if you isolate from the female embryos, you will get both X chromosome to be activated. Whereas if you have, if you isolate from prime, yeah, you get one X will be inactivated. Yeah, okay, so that's the difference. And then also you have localization of OCT4 activity. So one is at distal enhancer, and the other one is at proximal enhancer. Yeah, and then you also have a different distribution of trimethylation of lysine 27 on histone 3 protein, yeah, the H3K27 methylation. So you have 3 methylation site, yeah, distribution is different between the naive and also the prime, yeah. So you have for naive, you will get so it's direct. The trimethylation is associated with this, important because the trimethylation is associated with the downregulation of nearby genes, yeah, via the formation of heterochromatic regions, yeah. So you know when methylation happens, you get silencing of the gene expression. So they actually involve the polycomb associated gene promoters, yeah. So polycomb promotes the hypomethylation of the MVs near developmental gene. Yeah, so they are different. The distribution is different between the two naive and also the prime pluripotent stem cells. And lastly, they also express a different set of specific cell surface markers. So for naive, they express CD7, 75, 77, 130, and so on. Whereas prime, they have a different set of specific cell surface markers. Yeah, and also they involve different signaling pathway. Yeah, for naive, they involve BMP and also involve LIF. For prime, they actually need activin and model pathway to actually activate or express the core pluripotency markers OCT4, SOX2, and NO. Yeah. So when people trying to understand whether we can actually convert human prime to naive and vice versa, yes, people have due to knowledge of what is needed and what is not needed, what should be inhibited, what should be activated, and based on the genetic networking, the gene networking. So people have managed to come up with various methods that people can actually convert. Yeah, if you have naive from the early stage, yeah, and then you can make them to become naive, and then you can also make them to become prime. Yeah, and vice versa, the prime, particularly the human, yeah, so the human stem cells, which is known to be prime, you can actually make them to become naive with proper recipe and also inhibition and also activation of certain pathway. Okay, all right. So a lot of studies have been done. Yeah, so characterization method needs to be done once you claim yourself to establish an pluripotent stem cell line or embryonic stem cell line or induced pluripotent stem cell line, you really need to do characterization. So one of the characterization is you need to make sure they express pluripotency associated markers. They actually have the ability, so you can do a quick characterization by using in vitro pluripotency test by making them or direct them to form embryonic bodies. So embryonic bodies is actually a step where it's actually mimicking the embryos in the body. Yeah. So the embryonic bodies, they have to have quite a smooth boundary, let's say, and then they have to have this cavitation process to mimic the blastocele in the embryo. So you need to do this in vitro, quick. This is very quick to do. So this is actually from my group, yeah, the ABs, the embryonic bodies. So it's very quick to do. After day 1 or 2, you can get the ABs to form, and then upon day 4, 5, and 6, you can see the cavitation process happen. So you can see the apoptosis, the cell death here is actually mimicking the cavitation process that mimic the blastocele stage, the blastocele in the early blastocyst stage. Yeah. And of course, you need to confirm the pluripotency by using in vivo pluripotency tests. And also there are various ways of in vivo pluripotency tests. And you can also do, one of it is to do tritoma formation, when you actually directly inject the cell, the pluripotent stem cells into a new mice or immunocompromised mice. Then you can see they can form teratoma representing the three lineage, yeah, the three primary germ layers. Okay, that's why you get the teratoma to form. All right. So what about the application and limitations? Yeah. So we know that the whole idea of having pluripotent stem cell is because of their properties, they can propagate. Yeah, they can propagate. So you have a continuous supply. So whenever you need it, you have the stock, the part, in the spare part. So that's the idea. Because of they can continuously propagate, yeah, they can make more of themselves. And the other one, of course, the other properties is they can differentiate into a very wide differentiation spectrum. They can become all cells in our body, including the germ cells, yeah, including the germ cells. So you can do a lot of things. You can plan to do. Particularly, the ultimate aim is for therapy, yeah, to treat the disease and all. So you can grow them from patients. So iPSC, you can reprogram somatic cell from the patient, from a patient suffering for a particular disease. So you actually establish patient-specific as well as disease-specific induced pluripotent stem cells. And you can also get it from the early the blastocyst stage embryo, which is the embryonic stem cells. You expand them, yeah. So you grow them, you culture them, and then you differentiate them into whatever lineage that you wish them to become, to be used for various applications. Among all, you can use them for drug discovery, for disease modeling, yeah, to understand, you carry out fundamental studies, yeah. So then also you can do drug safety assays for drug toxicity screening, and of course for cell-based therapies. So these are the main applications of in general, the application of pluripotent stem cells. Yeah. So in specific, so you can actually grow. I mean, the specific application, yeah, as I mentioned, is for clinical and therapeutic applications. So it can be used for regenerative medicine. Particularly, you generate and then to replace the damaged or lost cells, yeah. And also it's actually you can also do gene therapy. You correct you establish the stem cell, and then you do correction, and then you get back the cells, transplant back the cells to the patient. So we call this gene therapy. And also you can use pluripotent stem cell for a cellular therapy. So now it's getting famous, but of course not directly from pluripotent stem cells, yeah, because pluripotent stem cells can have a risk of teratoma formation. So you need the best source for a cellular therapy or stem-free therapy, yeah, some people call it stem-free therapy. Cellular therapy the idea is to use this to use not the cells for therapy, Yeah, so that's why they call it stem cell free therapy. It's not that the therapy is free of charge, no. It's that it means the therapy we use therapy without using the cells and without injecting cells into our body. But what we are injecting is the secreted molecules, secreted therapeutic molecules from the stem cells into the medium. We collect the medium and then we use that therapeutic molecule residing into the medium as a treatment for diseases, for therapy. Yeah, so that's why it's called a cellular therapy. So a lot of people are now coming up using this, but mostly focus on mesenchymal stem cells. So if you were to start from pluripotent stem cell, you can. So you need to differentiate the pluripotent stem cell to become mesenchymal stem cells. Then only you harvest the medium that you cultured the pluripotent derived mesenchymal stem cells to be used in the cellular therapy. So this is what the medium that you collect is called. It contains a lot of secretomes. Yeah. So secretomes is all the, meaning that the secretory molecules that be secreted out into the medium. So you collect the medium. So the medium is called secretome because it's contained the secreted molecule. So that's why we call it secretome. So among the secretome, you have a population of cell called not cell, a population called exosome. Yeah. Sorry. So for clinical application, the APS, the pluripotent stem cells can be a source of cells and tissue for transplantation. Yeah. So a lot of people was wondering whether we already have how far have been gone, using pluripotent stem cells in terms of therapy. Yeah. As for now, we do not have any approved therapy using embryonic stem cells or pluripotent stem cells as yet. All are in clinical trials. A lot of them are in clinical trials. So I'm not going to go through in details of those. You can read this on your own, I suppose. So, but it's enough for me to highlight that there are a lot of clinical trials are going on using human embryonic stem cell derived differentiated cells, in particular for retina. Why retina is so popular? Because retina has confined, because the eye is very confined, yeah, so it doesn't involve much of other organs except for the nerve, yeah, so that's why, and also they're very easy to do the injection and stuff, so that's why most scientists decided to go towards eye disorders first, yeah, among all the staggered macular dystrophy and the blindness, yeah, so they actually, so all the macular degeneration, actually they target for clinical trials. I've gone through up to clinical trials. Yeah? Of course, we have also other clinical trials for other diseases such as Parkinson or heart failure, yeah, also spinal cord injuries, yeah, right. So this is the secretomes, yeah, the cellular therapy. You can get it from the mesenchymal derived pluripotent stem cells, yeah. For example, embryonic stem cell and iPSC. But of course, the secretome is famous for mesenchymal stem cells, yeah. So the mesenchymal stem cells, so once you differentiate embryonic stem cells and induce pluripotent stem cells into mesenchymal stem cells, so you can actually they can actually secrete all these valuable molecules, valuable paracrine factors that actually at the end of the day involve and enhance proliferation and differentiation. Yeah. All right. So there are clinical and also non-clinical applications. So the non-clinical applications, so they are actually what we get multidisciplinary. Yeah. So you need in order for us to apply pluripotent stem cell into clinical settings or into bedside application, you really need this to be happening. Yeah. So it's actually interrelated. Yeah. So non-clinical applications is meant for one, you can use it for disease modeling. This is the in thing now, organoid or in vitro model. So you use the stem cell, the pluripotent stem cells as a model. So that means because the idea is because the pluripotent stem cell, you can differentiate them into all sorts of cells, right? So you can make them, you can create a model, a disease model, or just a normal fundamental study, yeah, for you to understand. So you can do fundamental studies and then to model using the stem cell to model and understand normal and disease development. You can use a stem cell for drug discovery, drug screening, and drug toxicity, yeah, and also for biobanking. So this is the organoid, the 3D model, yeah. So the 3D model, people have because of the properties of pluripotent stem cells. So only pluripotent stem cell you can use only pluripotent stem cells to make your organoid. Organoid is a mini organ in a dish. Yeah? So it's a 3D in vitro organ models. Yeah. So it does not only mimic the what you call the expression of specific markers, but also it mimic the structure. Yeah, so it's mimic the cell organization and organ structure. Yeah, that's why I make it so valuable. Yeah, so it's a source from pluripotent stem cells. So you can use it to study developmental disease pathology and regenerative drug toxicity and so on. Yeah. So as for now, scientists have created a lot of organoids already. So among all the brain, yeah, the cerebral organoids, kidney, skin, retina, breast, and liver. Yeah. Right. What about utilization of pluripotent stem cell to tackle or to understand COVID-19? Yeah. So we all know that because people have discovered that you can actually try to understand. We need to understand in terms of the viral tropism of this SARS-CoV-2 virus. Yeah, that actually caused the COVID-19. So we know that the SARS-CoV-2, they actually the way it gain entrance is through the receptor ACE2. Yeah. So scientists have been wondering whether we can use pluripotent stem cells to mimic certain organs in vitro, and then we try to understand whether or not that particular organ or tissues has these ACE2 receptors that will gain entry, that will permit entry to this virus. So people have, using this technology, so people have come up with organoids, try and then differentiate pluripotent stem cells, human pluripotent stem cells, because you can't use human to actually check, right? No one will volunteer to do that. So you need an in vitro or a cell line that can use that can do that. So we use human pluripotent stem cells. We differentiate them into all the 3 primary germ-layer cells. So that includes pancreas, liver, endothelial, the heart, yeah, the macrophages, the microglia, the neurons. So the ones that are in 3D organs, they make them to become organoids, yeah, and then they actually check whether these organs and tissues actually express ACE. Yeah, so they know that the ACE are expressed. You can see all the red stain cells are actually the ACE. So we know that these cells are susceptible for SARS-CoV-2 infection. Yeah, and then they also want to check further using the organoids model, to check the cellular response to infection. We know that the cells, particular organs and cells, express the ACE2 receptors, but they also wanted to know whether or not they see response to infection, or they allow for infection. So they actually create an experiment for pseudo entry of SARS-CoV-2 virus. So you can see they actually certain cells actually were very susceptible for SARS-CoV-2 infection. Yeah. All right. What are the limitations of these pluripotent stem cells? Yeah. So limitations are one is we need to get the quality and safety issues must be fulfilled, so that we want to have a clinical grade human, so we can use it for bedside application. So one of it is you want to understand the cell division. Yeah, we want to understand the tetramer formation, and also we need to have a very good differentiation efficiency, so that there won't be any leakage. You know, once you differentiate the cells, you inject the differentiated cells, but if the differentiation efficiency is not good, it's not efficient, so there will be a leaking of undifferentiated pluripotent stem cell. Yeah, into that will cause teratoma formation. Yeah, and then we also need to, even though it's reproducible, yeah, so we know that the efficiency of reprogramming is not so efficient and it's quite slow. So we also know that the reprogramming, yeah, the reprogramming of the somatic cells actually promote certain expression of certain genes, yeah, especially the P53 or the P21 pathways that actually put a barrier to reprogramming. Yeah, so they actually lead to the apoptosis and senescence, yeah, which ultimately, the cell die. Yeah, because of that. So they will reduce the efficiency of reprogramming. We also need to come up with an idea where we have this independent integration. So we need to have a protocol where the transient independent, that we use small molecules, not genetically modified. It has been going on. All right, so application, I'm just going to share you. I feel like I only have like 5 more minutes or so. So I will share to you what my group has been doing. Yeah, so in terms of utilizing pluripotent stem cells. So my group focuses more on the embryonic stem cells, because embryonic stem cell is what I was trained for during my PhD. So, so one of it is we use embryonic stem cell. We use mouse embryonic stem cell to actually reveal and understand neural differentiation process. Yeah, so that we can get a better and high efficient differentiation process where we can get enhanced a better result of neurons that we generated in the dish. Yeah. So one of it is we use WIN1. Yeah, WIN1. So we want to check because during neural differentiation process, which mimic the in vivo development also, so we want to see whether we can at what time point we need to stimulate WIN1 in particular, so that we can get a very good high percentage of neurons, a good quality neurons, for example. So the idea is we will overexpress the WIN or stimulate WIN signaling by overexpressing WIN1 by so we need to turn on at a particular time point during the neural differentiation process, and we want it to make in a closed environment. Yeah, so we in that sense we use a Kryllox-P binary system. Yeah, so I'm not going to talk about Kryllox-P binary system now. That's just enough for you to know the Kryllox-P binary system is an inducible system where you can induce the expression of transgene, in this case WIN1, at a particular time point during a process. So now we actually stimulate WIN1 expression during early stage, late stage, and also constitutive expression. That means we turn on the WIN1 expression from undifferentiated state of embryonic stem cell, not during the neural differentiation process. So this is one. So overexpression of WIN1 we discovered, yeah, in mouse embryonic stem cell, and in early embryonic stem, in early EBs. Yeah, so we treat one of the time point is during formation of EBs, promoted the formation of postmitotic neurons. So you can see. So this is the during undifferentiated state, we promote the expression of WIN1, and then also during EBs. EBs is the early. Late means we promote them when they become neuroprecursor cell already. Yeah. So this is one example. We also use embryonic stem cell line to reveal the neuroenhancement effect of a natural product. I'm also working on natural product. Yeah. So we use a cell line, a transgenic embryonic stem cell line, which is 46C. Yeah. So 46C is a transgenic cell line where the GFP was locked into the SOX1 open reading frame. The SOX1 open reading frame is actually SOX1 is a marker for neuroprecursor cells. So in that sense, if that stage, if that cells is actually neuroprecursor cells, you got SOX1 to be turned on. Yeah. So SOX1 turn on indirectly, it will turn on the GFP. So that's why you can see here the green. This is embryonic bodies. So we have a neural differentiation process where we subject the stem cell to become embryonic bodies. Yeah. So you become embryonic bodies. So these embryonic bodies actually contains a SOX1 positive population. That's why they become green. So when we see green, we know that our neurodifferentiation process is successful. So and then we proceed, we dissociate this gene and then these embryonic bodies into single cells, and then we culture them and we promote them to become mature neurons. So this is mature neurons. So you can see touch1 is a marker for postmitotic neurons, CHAD is a marker for cholinergic neurons, TH is a marker for dopaminergic neurons. Yeah, so these are the neurons after you dissociate the cells, the embryonic bodies into single cell suspension and replate them on the PDL laminin coated plate. So you can see this is the morphology, and then you grow them and then you get this. Yeah, right. We also you can use embryonic. We also use embryonic stem cell line, the same cell line 46C, to reveal neuroenhancement of natural product by measuring the diameter of the neurite outgrowth. Yeah, the neurite. So these are the neurites. Yeah. So we treat with a natural product. Excuse me. Then we measure the diameter. So why is it important to measure the diameter? Because the extension of the cell bodies of the neurons and the specialized for receiving, processing, and also transferring synaptic inputs. Yeah. So the diameter of the neurite is playing a significant role. So it's actually significant element of neurons functionality. So that's why the bigger the size is, more or less they can communicate well and they become more functional. So the neurite diameter is increased when we treat with the natural product in this model. Yeah. So the number of neurite branches also increase when we treat with the natural product. So by doing this, so now that's how we utilize the embryonic stem cell to see the effect of a certain natural product. Yeah. We also create model, yeah, in vitro model. So this is cell line model for a particular type of disease. So one is we are interested in to see the beta amyloid peptide. Yeah. So we actually create our model, our ED model. So we call it more or less ED-like model. It's not 100% ED model, but ED-like model in the sense that they actually have increased the formation of ROS, yeah, the oxidative stress. So we treat them with amyloid beta peptide. So we use fragment 142 and fragment 25 to 35. So this is our neurons. So this is our neurons from the 46C, the embryonic stem cell line. So we treat them. So this is our result. So the treatment. So the model is successful. We managed to create the model. So at the end, you can see the upon treatment with the AB peptide 142 and 25 to 35, they increase the production of oxygen, ROS. Yeah, they are oxygen reactive species. Okay, so using this model, so we conclude that the AB induced toxicity in culture of neural cells, yeah, differentiated from embryonic stem cell to degeneration of ROS. So this is very preliminary, yeah, right. And then we also use mouse embryonic stem cell line to further do, but this is to generate a model for in vitro oxidative stress model for neurodegenerative diseases. But this model is using stem cell line, yeah, as opposed to normal model cell line that people use. Normally people use cancerous cell lines. For example, the human neuroblastoma. To most people use human neuroblastoma, the Shishi cells, to actually create the model to do neuroprotective or neuroprotective or neuroenhancement, neuroregenerative. So now we are trying to see whether we can use embryonic stem cells to create the model. So we managed to create the model. So this is the cell line. Yeah, we managed to differentiate into all types of neurons. So we treat them with hydrogen peroxide to make them oxidatively stressed, and then they die, of course. And then we treat the cells with the So we managed to see, so these are the model that we created, yeah. So this is the model that we created. So it has increased the ROS. Right, and then we also reveal neuroregenerative using the model. Yeah, so this is neuroregenerative. So we treat the cells with, because we want to see the effect of neuroregeneration. So we damage the neurons first. So this is how damage the neurons is. This is, then we treat with the natural product to see whether they can regenerate. Yeah. So then this is the result after we treat the damage. This is, you can see this is very damaged. Yeah? So we treat with hydrogen peroxide, the neurons are damaged. And then we treat with natural product and to see whether they can get back. Yeah, they can regenerate. Yeah, it's regenerate. So you can see they can regenerate. You can see the number of apoptotic cells are actually reduced. Yeah, and also the we also calculate the average neurite length. So they also increase. Yeah, they also increase. Not so much as the DP as our control, but it's better than the damaged neurons. Yeah? Okay. We also use neuroprotective. Yeah? So that means now, just now regenerative. So we damage.
Sorry to interrupt, Prof. So we have another 2 minutes. Is that all right?
Okay,
To
right.
continue?
Yes,
Yeah.
yes.
Thank
Okay,
you.
I will wrap up. Thank you. Sorry. And then we also do neuroprotection, neuroprotective effect of curcumin. So here also we can see that they are actually using the model, we can see the effect. Yeah. All right. Okay. So now we know that one of the ways is we need to know how to expand the cells in vitro. So we need to scale up and also to use in a defined or xeno-free medium so that it can be used in transplantation. Yeah. All right. So gaps in pluripotent stem cells. What can we do now? So you need to understand the mechanism. So you need to understand and define the naive state, and then you need to get it optimized. Yeah. You need to know which pathway needs to be inhibited, which needs to be. I mean, all these have to be at optimal level. And then you need to enhance the cell renewal, yeah, and also the differentiation. So these are the things that you can do, we can do, yeah, in Malaysia. So we can actually create our own kind of stem cells, pluripotent stem cells, and then we can do our own kind of, we can try to enhance cell renewability and also try to come up with efficient in vitro differentiation assay. Yeah, so now we can do biometric support and stuff. And so you can also do and come up with biobanking. Yeah, so we can also get a robust and reproducible protocol for organoid. Yeah, so efficient derivation of organoid. You can do also biobanking. Yeah, so biobanking, especially for low passage GMP grade human pluripotent stem cell for stem cell banking. Yeah, so for our population, for our cancer. So those who are interested in mostly in iPSC, they should do come up with try to generate induced pluripotent stem cell lines and then try to screen for HLA, so we can come up with biobanking so that we can use the cell lines when it's needed by a particular compatible individual. Yeah. Okay, so this is, I'm just gonna have a few seconds to actually tell what my group is doing. So my group is called Stem Cell and Neurodevelopment. So we are in UPM. So this is what I'm doing for my work, yeah, for my team. All right, so I would like to acknowledge all the people who have been working. These are my students, as most of them are graduated now, most of them. And then I want to acknowledge my collaborators from UITM, and then from University of Edinburgh, and also USM, and the rest are from UKM. And my funders, NLGS, FLGS, and GRF for my students. And thank you very much. And I apologize for exceeding the time limit given. With that, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Associate Professor Dr. NORSHARIZA binti NORDIN, for his very holistic introduction to what is pluripotent stem cells actually. So we have a complete understanding for what is pluripotent stem cells and what are the different types. You give introduction to the differences between the naive and also the prime state of pluripotent stem cells. And we also did discuss about the epigenetics and how we can transfer or manipulate this information into a research activity. So you have come out with a neurodegenerative potential of embryonic stem cells, and also you are interested in the secreted molecules from these pluripotent stem cells. I think this talk already summarized most of the important parts and also addresses the essential issues regarding pluripotent stem cells. Thank you very much for the talk just now, Prof. NORSHARIZA. We will keep one question. We got one question for you, but I hope you can keep it for towards the end. Is that all right? Okay, so it's in the comment section. We will answer this question towards the end. All right. So Prof. NORSHARIZA already discussed about the fundamental knowledge, how we can differentiate the cells, pluripotent stem cells into various lineages. And we have in our body, we have the somatic cells. Okay, we have also the germline lineage. So with us today, we have Associate Professor Dr. Muhammad LOKMAN, that going to talk about the advancement or the application of stem cell technology, particularly in reproductive biology. Let me introduce our second speaker for today. Dr. Muhammad LOKMAN received his BSc in Biomedical Science from UKM, and after that he continued with Master in Pharmacy in the same university. And he did his PhD in human germ cell development and spermatogenesis from stem cells in University of Sheffield, United Kingdom. He is currently holding a position of chairman of IIUM Molecular and Cellular Biology Research Cluster, and he interested in the molecular reproductive biology and also cellular technology. Without further ado, the floor is yours.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, Dr. Tengku Faris, for chairing this session, and also for the first speaker, Prof. Dr. NORSHARIZA, for completing the everything about the stem cell, pluripotent stem cell. Very interesting. So now I would like to share with you some kind of activities that our group have been done here, that we are more focusing on reproductive stem cell, okay, the gamete cell. Okay, let me share with you the slide first. Wait a minute. So can you see my presentation? This one is. Let me share first. Tengku, can you see that? Share screen. Okay. Yes. Can you see the presentation?
No.
No? Wait. Wait a minute. So mana pergi dah ni? Transfer. Dah share dah? Just click that. No. This one. Tak dapat.
Are we having some issues there?
So we try to share the slide. Wait for a while. Because this is normally we use the Google Meet kan, but this one is, let me I try again, yeah. Is it video file or?
It's not the video file, it's the share screen button.
Share screen, yeah, it's supposed to be. Okay.
I think it's working now.
Is it?
Yes, yeah, it's appearing now.
It appears. You can see that, yeah?
Yes, I can see. Yeah.
Okay, cool. Right, you can see this one?
Yes.
Advancement of stem cell, yeah?
Correct.
Okay, cool. Thank you very much. Okay, sorry for the technical thing. Okay, so my talk today is the advancement of stem cell technology in reproductive biology. Okay, yeah.
Dr. LOKMAN, maybe you want to blow it up a bit, maybe full screen. Can you do that?
Yeah, I did the full screen already.
Oh, it's showing the sidebar for the PowerPoint too, but that's fine. I think it's just like a bit smaller as compared to the normal.
Yeah, I'm not really sure how to, but I think should be fine then. Yeah. Okay.
Yeah.
Okay. So when we're talking about the gamete cells, okay, we have to be careful because of they are haploid cells. Okay, that's the most important things and the most crucial and tedious part to ensure that we can able to differentiate the pluripotent stem cell, either induced pluripotent stem cell or embryonic stem cell, to the gamete cells. Yeah, gamete means sperms or ovums. Okay, but I'm more focusing on sperm cell actually. So if you have a look here, they have a several from the PG cell, okay, primordial germ cells. So we can have, at the end of the day, the baby will have either testis or ovary. So there is no intermediate, or there is no in the middles, okay? So they will be purely testis or purely ovary, okay? But nowadays, we're talking about the transgender and so on, yeah? Okay, so we have germ cells, and then we have type A spermatogonias, and then so on until we can get the spermatid and then spermatozoa with the spermiogenesis, yeah. And then if you look at to the circles of the ovum itself as well, they will produce one kind of the ovums, okay. So this is actually our approach previously, okay. We can get like, yes, we can get embryonic germ cells, and then we can get spermatogonial stem cells from the adults, and then we can get the embryonic stem cell from the embryo that we discussed with Dr. Shariza just now, okay. And then we have the fetal developments and neonatal pubertal adults development. So we have a different types of the stem cell that we can get along the way of the spermatogenesis, okay. So we can get the fetal development, we can get the PGC, the germ stem cells, and then we can get gonocytes. We need to get the gonocyte, which is still the diploid, and then spermatogonia, which is still diploid, and spermatocyte or spermatid, which is already haploid. Okay, there is a review from one paper related to the attempts of several researchers to develop or to differentiate the stem cell into the ovums. Yeah, if you have a look here, we can have several attempts already have been done to provide or to get the ovums. Yeah, but the sperm is, because of the sperm, we need to have the spermiogenesis that a bit triggered on this. Okay, we need to have the really forms of the sperms. But nowadays, in order to have the IVF technology, we don't need to have the real forms of the sperms or the spermatozoa, but it's like the round spermatid should be enough. Nampak tak? Wait a minute. Can you see now?
No, we are losing your slides.
Okay, wait a minute. Hmm? Hmm? Tak boleh pakai yang tadi je. Tekan ini. Hmm? Look. This one? Nampak tak? No. You can see now? No.
No.
Tengku? No,
No.
wait. No.
The previous one is okay.
Yeah, I think we just wait a minute. This one, just follow this. Masuk sini. Security and privacy. Which one? No.
If you use the Apple device, maybe they block some features. So you go to the privacy and then you need to unblock it, or maybe allow them.
Security and privacy kat mana? Tak nampak. This one. Privacy. Screen recording. Sekejap. Boleh? Okay. Right. Sorry for this this kind of
because these using the different platform, I think this time, so maybe you're not familiar with these features.
Yes, because the Apple as well. Okay.
Yeah.
Right. Okay.
Now I can see the slides.
Yeah. Okay. We go. Right. This slide's not going to be very big for me. Okay. Okay. Next slide, please. Go on. Okay, this is like no. The other slide. Okay, earlier. Go back earlier. One more slide. One more. Okay. Yes. Okay, that's it. No. Okay. Next slide. Next slide. Okay. Okay. So this is like the way we can get the sperms. No, next this slide. Okay. The sperm. Okay, and then we can have like different markers. Okay, different gene markers that we can have a look. Okay, in order to see that the cells were growing to the right pathway. Okay. Next slide. Okay, this is like several gene markers that we identify to get the sperm and ovums from the different stage of the spermatogenesis or oogenesis. Yeah, so from the PDC, genocyte, spermatogonia, meiosis, and spermatocyte. Yeah. Same goes to the ovum as well. So this is like several markers that maybe we can transfer into the pluripotent stem cell in order to see that it will flourish the cell manage to differentiate into the right pathway. Next, okay. So this one is like the hypothesis that because we're trying to have like a different ingredient in order to get the right or the suitable condition in order to get the differentiation happens. So we try to because after several reading we got that retinoic acid is something that could lead the differentiations of the gametes itself. Okay, next. Okay, there is several attempts that we did. It's like we tried to do combinations with the retinoic acid with a certain like anti P53 and so on to ensure that the cell will keep on growing, and then with those kind of attempt we can get the optimum condition that suit to the differentiation process. Next. Okay, this is what we got previously. Is the we used the fish, okay, fluorescent in situ hybridization to ensure that the cell are in the phase of the haploid cells. Okay, so the cell, if you can have a look here, the chromosome supposed to be double, okay, but then when we have like only one blue, one green or one red of the cell of the colored chromosomes, so we can say that this cell is become haploid. They turn from the diploid to haploid. So this is like this has happened because of the condition media that we use, and normally the population is very, very small. It's about less than 1% in the whole population. Normally, what we do, we're not introducing or we're not approaching the embryo body system, but we are doing the 2D or two-dimensional or cell will be seeded into the plate of the cell of the flask. Okay, next. Okay, this is like examples of the stained positive cells that we can get from the activities. Okay, I just show you in very fast. Okay, next. Okay, next. Okay, so this is like because when we do the spermatogenesis process, we want to follow the real physiological process. Okay, so we have a several step. First step, we want to ensure that the cell is like entering the pathway, and then after that we try to change the media in order to ensure that they are entering the spermiogenesis or half the chromosome numbers. Okay, so that we tried the attempt is like we want to have like the real like serum condition that we attempt at that time we attempt to have the neonatal mouse testis at age of 10 days, so that we want to have this kind of the conditioned media to ensure that the cell will go and behave or differentiate correctly. Okay, this is the example. Okay, after we have a several day, and then we prolong the culture with the new media or new condition media. Next. Okay, this is how we at first we try to attempt that we cut the epididymis of the neonatal mouse testis, and we try to inject the cell into the epididymis neonatal mouse testis, because physiologically the sperms the formation or the maturation of the sperm happen in the pipes, like pipeline something, so that it's supposed to be, we'll be thinking like we need to mimic this kind of physiological activity in order to ensure that the formation is correct and the maturation is properly done. Okay, so what we do is we try to inject the differentiated cell into the epididymis of neonatal mouse testis. Okay, and next. Okay, so this is like the attempt that we can do. Okay, we managed to get the haploid cells, and then we further using the neonatal mouse testis, and then we can get some markers of the late differentiation, a late spermatogenesis of the cell. But at the end of the day, we not managed to get the sperms as a spermatozoa forms as it is. Yeah. Okay, so this is something that attempt that we have done previously, and then what we do now is we are focusing no longer on human because of there is a lot of ethical and anything related to the issue, but now we are focusing on endangered species. Lately, we as you know, as you're aware that we lost our Sumatran rhinos, okay, but fortunately our group managed to derive the cells, okay, managed to derive the fibroblast like cells of the from the different organs of the three last Sumatran rhinos, which is Iman, Kertam and Puntung. Okay, so next. So okay, this is like the sperms that we collect in our system. And firstly, I would like to tell you that we having the cell biobanks, or we call it frozen zoo, to the endangered species in our cluster building, and then we have several kind of sperms, fibroblast cells and tissue and so on that we bank into this frozen zoo. Okay, so next. So this is the cell that we managed to derive from the Puntung, Kertam and Iman, the last three rhinos that we have. Okay, so the cell is growing well in the good condition now, and then we are now into the attempts of cloning of this kind of cells. Next. And we managed to do the karyotypings of these cells because of it is very tedious, because of we have 46 but they have 84 couple of chromosomes, 84 of chromosomes, so we need to have like 42 couples of the chromosome to do the karyotypings. But this one is for sure is not being validated because of so far we try to find the papers published related to the Sumatran karyotypings chromosome is not yet appeared, yeah, because of we need to really focus on this kind of research, because of not many doing this kind of research. This is related to the endangered species. Okay, so I think in my last slide is I want to, yeah, what we are doing now, the possible application, next please, is related to the, like what Dr. NORSHARIZA say about we can do the induced pluripotent stem cell with the fibroblast that we have, okay, and then we can get the embryonic stem. We can get the pluripotent stage of the cells, and then we can turn back into sperm and ovum, and then maybe we can just do the fertilizations, and then we can put in the surrogate moms, or we can directly using the cloning system, somatic nuclear transfer system, so that we can have like the ovums, the enucleated ovum from maybe from African rhinos or something that we can get from the zoo, and then we can transfer the fibroblasts into that nucleated cells, enucleated ovums, and then they will like start to differentiate in the forming the embryo and so on, and then maybe we can get back our own rhinos that we lost last year. Okay, and of course the genomic database should be because of when we jump into this field, there is a lot of problem that we face because of the genomic database is not being completed for the Sumatran rhinos. So that's why we are having problem in producing like monoclonal antibody and so on in order to get the precise or to confirm the cell that we got. Okay, so I think we have to work together in order to get like to settle down the database and so on, so that will be smooth our activities. Okay, so I think this is like a sharing moment to all of you, what we have done and what actually the possibility. And of course, if we try to do like cloning and so on to the human, we will be having the problems because we are not allowed at all to do the reproductive cloning. But for the treatment cloning, or we call it, I forgot the name. Okay, it is still allowable. Okay, but for the reproductive means that we clone the whole organism is not allowed to the human. Okay,
The therapeutic, I guess.
Therapeutic, yes. The therapeutic is okay, still allowable, but not the reproductive cloning. So I think I end up this talk, and I'm really sorry because of these technical problems. And I hope you can see it clearly, my slide. So thank you very much, and Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Associate Professor Dr. Muhammad Lokman, for a very nice introduction on the advancement of the stem cell technology, especially in the reproductive point of view. Maybe some of our attendees cannot see clearly the slides, but the Secretariat will provide it after the session. Thank you very much, Dr. Muhammad Lokman, for the application of the stem cell research in the reproductive biology. Let's move on to our third speaker. Let me introduce our speaker. She is Dr. NURUL NASIM binti Muhammad YUSOF. She is currently a research officer in the Hematology Unit, Cancer Research Center, Institute of Medical Research, National Institute of Health, Malaysia. She did her BSc in biotechnology in University of Manchester. After that, she continued in stem cell technology in University of Nottingham, United Kingdom. And after that, she proceeded with her PhD in human development, focusing on stem cell in the same university, which is University of Nottingham, United Kingdom. Today, Dr. NURUL will discuss about the cardiomyocyte. We want to see how is the process of pluripotent stem cell differentiation to the cardiac lineage. So without further ado, I would like to call upon our honorable speaker, Dr. NURUL NASIM binti Muhammad YUSOF, to deliver her talk. Please welcome.
Hi, Assalamualaikum warahmatullahi wabarakatuh. Thank you, Tengku Faris, for inviting me. I would like also thank you, Prof. Shariza, for introducing a little bit about the non-clinical side of human pluripotent stem cell. So let me share my screen. I think I have problem sharing too, Faris. I think maybe the platform.
So in the
Yeah, in the share, in the down button share, right? So I have
yes,
clicked the share screen. So it has your entire screen, application window, and the Chrome tab. But when I want to try to click share, I couldn't click the share button.
are you using the Apple device or is it
I'm using Windows.
is it a Firefox or Chrome
It's a
browser?
Chrome.
It's supposed to work with Chrome optimally, but maybe you can refresh the
Wait. Oh
screen.
yes, yes. I think now
Yeah,
it's working.
it will take some time, I think. Yeah, I think it's appearing now. Yeah, you got it right.
Okay, is it working now?
Yeah,
Yep.
yes.
All right. Okay. Thank you. Assalamualaikum everyone. My name is Dr. NURUL. I'm from Institute for Medical Research. So today I'm presenting about the unleashing the potential of human pluripotent stem cell derived cardiomyocytes, HPSC-CMS, for disease modeling and drug safety. So just make sure you know this abbreviation because I will keep on mentioning it. It stands for human pluripotent stem cell derived cardiomyocytes. So if you see the headlines of the current news recently, you will notice that they are in the same topic, that drugs now is become more expensive and the new chemical entity has been discovered at a lesser amount with the expensive price. So the drug research is expensive and the cost is rising. For example, if you see in the 1950s, between 1950 to the 1960s, at the 1 billion spend, we have around 70 drugs being produced in the market, which means like one of each drug is cost around 14 millions. But over the past 6 decades, the ease of discovery has been difficult. So you can see in the 2015, like 1 billion has spent to develop one drugs only. So a lot of money has been invested to make sure that the drugs is working. But apparently, when the drugs has put into the human, 9 of the drugs has failed. And of course, that one drug has made into the market has to compensate all the costs of the failed drugs along the way. So why?
Your slide is disappearing. Maybe you can share back. Yeah.
Okay, wait. Can you use? Wait. Can you see now? No.
No.
Kejap eh. Share. Stop. Wait. I will stop sharing first again. Share screen. Share screen. Can you see anything now, Faris?
No.
No.
Oh, now it's loading now. Okay.
Okay.
But can you blow it up, like make it like a full screen? Is it possible?
It's already full screen now.
In your screen is full screen, yeah?
Yeah. In my screen, it's full screen. Okay. Macam mana? Kejap. Wait. I will stop sharing again. Let me try. Wait. Sorry for the difficulties.
So when you sharing, there's a 3 options: screen, window,
Yeah.
and Chrome tab.
Yeah.
Okay,
So
you choose the window one.
Okay, yes. Okay.
And then we show up the whole screen for the participants.
Is it there?
Perfect, yes. So now maybe you want to click F5 button, maybe?
Okay. So is the screen full now?
With the sidebar for the PowerPoint.
I already make it, sorry, I already make it
Okay.
a slideshow, but
That's fine. Yeah, that's fine.
yeah.
Mhm.
Let me try again. No, did you see the full screen?
Yes, yeah,
Or with the slide
with
on the
the
side?
sidebar. But if you uncheck the bottommost at the note section there, the button, you know, the glass, you know, at the bottom sidebar, just click it on. Yes, on the right, yeah, it will disappear. All right,
Okay.
it
Is it
become a
okay
bit
now?
more, okay. Yeah,
All
yeah,
right.
it works.
Okay. So should I start again? Sorry, I'm not sure where did I stop. Okay, so I start again with the first slide, okay? So the first slide, I would like to mention that the drugs research is expensive and the cost is rising. So compared in the 1950s and 1960s, per 1 billion of the money spent to make a drug, we can out to the market like 70 varieties of drugs. But now with 1 billion of drugs, we can only out of the market only one drugs. So interestingly, out of 9/10 of the drugs has failed along the way. And of course, this one drug has made into the market has to compensate all the cost of the failed drugs. So that's why the cost of the drugs now is really costly. So why does this happen? So if you see the current drug development pipeline, it always starts with the discovery, which is with the in silico model, cell line, and animal model. Proceed into the preclinical testing, next to the phase 1, phase 2, and phase 3 of the clinical trial, which is in human, before it's been approved by the FDA or the European market. So if you can see during the phase 2, the safety failure rates are 22%, and the phase 3, in the safety failure rate is 35%. So why we can see that the failure rates are increased from the 22 and 35%? They are same. Phase 2 is in the human and phase 3 is also in the human. The only difference is because phase 3 is a longer trial. So you can see that the drugs that fail most is during the chronic time and also in a larger population number of patients. That's why we can see more drugs fail. Taking together, it takes around 12 years to make one good drugs into the market. And 85% of the new drugs fail in the phase trial in human. It's not something surprisingly, we know about that. So why does this happen? So most of the drugs that fail is due to the unexpected cardiotoxicity. Well, it's about 28% of drug withdrawn because of the unexpected cardiotoxicity. Cardiotoxicity is very complicated. You can have a heart failure, you can have atrial hypertension, you can have myocarditis, pericarditis, thrombosis, pulmonary hypertension, ischemia, and also arrhythmia. Sorry. So why does this happen? Okay, so why does that happen? Is it because we don't have inaccurate model? Is it because this drug has been tested on the healthy people only? They didn't test it on the individual that have the mutation. They didn't test it on the individual that have variation, or they didn't test it on the individual that have polymorphisms. Especially for the polymorphism, it's really tricky because with the mutation, you know it's between the disease and the healthy, but in the polymorphism, it's a spectrum. So among the polymorphism, it is CYP2D6, CYP2D9, beta-2 adrenergic receptor polymorphism. And also, the most important thing is it because we don't have unsuitable platform to detect this cardiotoxicity in the drugs. So in the preclinical, we know that the current model has been tested in the animal model. So everybody know that human and animal is not the same. For example, the mouse and human is not the same. You can see the bit rate from the mouse is 600, but the human, the bit rate is only 60 bit rate, approximately around 60 bit rate per minute. So we might think that the bit rate is the major problem, but actually the bit rate is not really a problem. The problem is the biology behind it. So we can see in the animal model, behind the bit rate, there is like the electrical mechanism or the electrophysiology that make the peak and the peak, the tracing and the timing or the beating different between the species. So you can see in the mouse, it beat at the 10 hertz per minute, and on human is 1 hertz. In the animal, we can argue that although it has the identical ion current to the human, but there is difference in the bit rate, meaning the electrophysiology also is different. In the larger animal, the larger the animal, you can see is more similar in the electrophysiology and the cardiac contractile compared to the human. But then due to the ethical reason, we cannot test all the drugs in the larger animal, because it's going to be too costly to maintain the larger animal compared when you conduct it in the mouse or rats. So what should we do now if animal is not suitable? Well, we can use human, but then we need to remember that human and human is not the same. Like me and you, we are different. It's not like when we try to do a skin allergy test. With skin allergy, it's easy. We can just put some kind of antigens in our hand to see like which red spot appear to me and which red spot appear to you. But with the cardiotoxicity, you can't do that. You cannot take someone's heart, take a small piece of them, and then expect it to grow, and then you cannot compare between this heart. But then you can do it using the human primary cardiomyocytes. So, I forgot to mention that between human, there is a genetic polymorphism or individual variation across the population. So when we do the drugs developments, not all people is included in the population. Usually they will only choose a healthy person, not people with the rare disease, not people with have a mutation, not people with have a specific polymorphism which only exists probably like 0.1% in the population. Yeah. So, okay, with the human primary cardiomyocyte. So what is human primary cardiomyocyte? Human primary cardiomyocyte is taken directly from the heart. Then it will homogenated and enriched to become a small population of cardiomyocytes. But then the problem with the human primary cardiomyocyte is the availability. You can only have it mostly commercially, and it's going to be expensive. I'm not sure if you can make it on your own, because you need to have a consent from the individual after they died. So the harvesting time to collect this primary cardiomyocyte is really time consuming and labor intensive. You need a special operating theater and a short time to make sure that this cardiomyocyte is really viable. They also have a limited lifespan. They can only survive only a few days, and also low throughput, meaning you cannot do much experiment with them. And even after hours of the isolation, they have altered contractile machinery even at the physiology of the 37 degree. For example, you can see the disappearance of the T-tubule of the heart, and also some other important structural which will be disappear after the isolation. So this human primary cardiomyocyte also is not preferable for experiment, because they have problem with the low adhesiveness. They cannot be co-cultured with other cells. They not stick properly into the flask, and which make them unsuitable for the disease model, because they are at the their terminal state and also at their limited quantity. So what is the solution for the mutation or polymorphism or rare disease in the human? So we can take the advantage of using the CRISPR-Cas9 to generate human pluripotent stem cell isogenic variants. When I say the isogenic variants, you have to make sure that all the background is the same except the point of mutation or the point of polymorphism. So this is the example of my previous work in my lab in Nottingham. So with the help of my postdoc, Dr. Alexander Kondrashov, we have been developed a 4 isogenic line that coming from one parent line. For example, I will just make it short as a GE. So from the GE, we can use a CRISPR-Cas9 with a specific gRNA and the specific targeting vector to insert a specific DRB locus into the first parent line. So the first parent line is GE. We make it into a second line which is GQ, RE, and RQ. So this GQ, RE, and RQ, they are all have the same background, exactly 100% the same, except at the point of the variance. So when you have the one mutation only, or one polymorphism be edited, you can focus. Anything that come out of the difference, you will know is coming from these changes, not because of other genes, not because of other mutation, not because of the comorbidities. Wait a minute. Okay, so after you edit the CRISPR-Cas9 to make a specific edited human pluripotent stem cells, you can differentiate them into cardiomyocytes. So by studying, one of my colleague in Nottingham has studied the embryogenesis of human development and come out with a specific Nottingham cardiac differentiation protocol. So by using the protocol that mimic the embryogenesis, we put up a small molecule. We put up a growth factor BMP4 and activin A together with the small molecule KY and also ZAV to make it into cardiac inductions. So after 2 weeks of cardiac differentiation, we can have a beating cardiomyocyte. So along the way, you can see that each variance, GE, GQ, RE, and RQ, although they are in the background, they are the same, but when you do differentiation, how they react in the differentiation on the day 0, day 2, day 4, day 6, they are all different. But then in the day 12, they are all beating. To make sure that this is really cardiomyocyte, we have done the immunostaining by using the alpha-actinin, the green co-stain with the nuclear DNA, which is DAPI. And this alpha-actinin is the characteristic of the cardiac muscle striation. So we can see from our Nottingham in-house differentiation protocol that the percentage of the cardiomyocyte, the purity is above 80%. And to make sure that all other further testing is specific on cardiomyocyte, we only choose when the efficacy of differentiation is 80%. For something that is below 80% efficacy in differentiation, we will not proceed into any drug testing. Okay, so this is the summary. For example, what I have done in Nottingham. So this project is funded by the BHF, the British Heart Foundation. So I have genome edit the beta-2 receptor to study the functionality of the beta-2 adrenergic receptor polymorphism. So as I mentioned before, I have created the 4 set of isogenic variants harboring beta-2 polymorphism at the position 16 and 27, which were introduced in the parents line of the GE, which also known the West7, the embryonic cell line. So the edited isogenic control has successfully been edited. We have done sequencing to confirm it. And we also have to check the pluripotency, whether it still retain their pluripotency or not. And they are still retain the potency. You can see from the NANOX 2 or 4, and also the karyotyping is normal. There is no mutation occur. Next, we have checked the cardiac differentiation capability to make sure that when we do all our phenotyping, we can do it on the cardiomyocyte. So we have generated into a cardiac differentiation. So when it's become a cardiac differentiation, we have checked the beta arrhythmic profiles. And also we have done the functionality assay. For example, in mine, because the beta arrhythmic is the important polymorphism that play the role in the cyclic AMP. So I have done the using a functionality using the pGlow20F to check the glow sensor activity, where you can quantify whether the cyclic AMP, or you can check whether the desensitization or the phenotypes of the GE, GQ, RE, and RQ is different or not. So now I think you might be eager to know like how the beating cardiomyocyte look like. So this is example when they are not that dense. I hope you can see the beating. If you cannot really see its beating, it's okay, no worry. So I will put the bigger one. Okay. That's how our in-house protocol using the Nottingham cardiac differentiation protocol. That's how our cardiomyocyte look like. Again, the pacing is really smooth, it's synchronized. But then this monolayer cardiomyocyte still have its limitation. So one important limitation is this cardiomyocyte is still on its immature state. It's not like adult cardiomyocyte. The phenotype is still like a fetal-like cardiomyocyte. It is still immature. So how to solve the problem about the immature cardiomyocytes for the drug testing? So this is one of the example that can make the cardiomyocyte more mature. Rather than the 2D, the monolayer, we can design it to be the 3D. So this work has been done by my collaborator. This is developed by the Thomas Ischenagel lab in the Hamburg, Germany, together with the Ingramin heart. So they have developed the 3D engineered heart tissue. I hope you can see them flicking, beating. So what they do is they take the 2D monolayer cardiomyocytes, they cast it into a fibrin hydrogel, which held between the two BDMS posts, and then the deflection of the post is quantitated by the video optical recording to allow the contractility monitoring. So you can monitor the contractility of the heart based on the deflection of the BDMS post. But also you can introduce a transgenic plasmid into them. So what they do, they have incorporated a transgenic encoded calcium indicator, which is the GECI CCAMP66F, for the assessment of the calcium handling in the cardiomyocytes. So the outcome, it was mentioned that this EHT is more mature phenotype due to the mechanical loading by elastic PDMS post, and the contraction was under the autotonic state conditions. Okay, so we know we have a 2D and we have the 3D. So what's method currently be used to quantify this cardiomyocyte? So there are three important elements to quantify the cardiomyocyte. You can use a beating motion, for example, like the impedance assay, atomic force microscopy, traction force microscopy, and video microscopy. And also you can detect based on the intracellular ion change, which is the patch clamp, which is really invasive and really hard to do. And you can also do the fluorescent imaging or multi-electrode assay. And then you can check with the intracellular ion reflux, which mainly use a voltage dye and then which been detected by the fluorescent imaging. So this is the summary of what method has been used to detect the electrophysiology and contractility of the cardiomyocytes. Each of them has a pro and a cons, which I will not go further. So in my experience, I have given the privilege to use the cell-optic platform, which created by the Godfrey Smith from Clyde Bioscience. So Clyde Bioscience is a spin-off company from the Glasgow University. It's been created by scientists which know the urge to have a system to detect the contractility, calcium, and the voltage. So this is the machine look like. This is more simple. It's actually more complicated. And this is if you want to trace the voltage, you can see the voltage coming out in here. And with the cell optic, the most important thing, you can pace them according to the electrical pacing. For example, the beat rate of the cardiomyocyte is spontaneously beating at 1 Hertz. And then when you try to pace it at 1.2 Hertz, you can see that the cardiomyocyte will follow the pacings. And then when you increase the pacing into 1.5 Hertz, again, the cardiomyocyte can follow the pacings. So what this cell optic actually does? It is a multi-parametric medium throughput assay platform. It can record the contractility using the high-speed camera, which it records 1,000 frames per second to measure the contractility based on the bright field imaging. And by using an algorithm, it can quantify the contraction time and the relaxation time based on the contraction peak. And it also can measure the electrophysiology response of the voltage via the fluorescent measurement. For example, by using the voltage sensitive dye, it's either dye for ANIMS or you can use flow volts. And you can also measure the intracellular calcium measurement by using the FURA2 or the FURA4. So this is the example of when we test our drugs, nifedipine. So nifedipine is drugs that have been used to cure blood pressure and also chest angina. It is known as a negative inotropy because it reduces the contractions. So you can see compare with the baseline when we treated the cell with the nifedipine, we can see the contractility has been decreased. Also with the voltage, you can see the APD, the action potential duration, has been decreased when we treated them with the nifedipine. So this platform can be used to quantify the drug's effects on cardiomyocytes. Another system, I forgot to mention, that's in the cell optic, you have to do everything manual at the sequentially. So it's kind of time consuming and also tedious. So I used to read half of the 96 plate of cardiomyocyte, and it took me like 1 whole day to read it. And this machine is only currently available 3 in the world, 1 in Glasgow, 1 in Nottingham, and 1 in London. And only a few people know how to use it, and I feel privileged to have the opportunity to use this machine. So the next machine that can use to quantify the cardiomyocyte is the triple transient measurement, TTM, which has been developed by the Baron Van Meers and the Christian Memory Group. So it's almost similar with the cell optic. By using a certain dye with the fast microscopy, you can record the voltage, contractions, and also the calcium peak, just by using a different dye. For example, for contractility, it will based by the cell membrane level, the cell mass deep red, and with the voltage tracing, you can check using the dye anin 6+, and with the calcium sensitive dye, rod 3, you can check for the calcium peak of the cardiomyocytes. But then the limitation of the cell optic and the TTM, both is being conducted outside the sterile condition, and it is terminal, meaning you cannot do them under a chronic exposure to the drugs. You can only do it a short time, for example, like 30 minutes after exposure to the drugs only. But with the drugs development, you know how it's important to do the drugs at a prolonged time, especially a chronic. So you have to make sure that this cardiomyocyte is sterile. You don't want it to get infected, contaminated, because it will yield a different kind of results. So here come another one nice machine that I really like to use. It's an Excelligent RTCA Cardio ECR, which has been developed by the Asia Bioscience. So this Excelligent RTCA Cardio ECR, it can allow measurement of the chronic effects, like I mentioned, because it's so small. You can just put them inside the incubator to measure the tracing cardiomyocyte. So it's based on, unlike cell optic and cell optic and TTM, which is based on the microscopy, this RTCA Cardio ECR is based on the impedance. It's measure the contraction based on the impedance, and it also measure the field potential, which means the ion channel activity of the cardiomyocyte, and it measures simultaneously using two different kind of electrode. So this is example when we test our drugs, the acetylsalicylic acid, also known as the aspirin. So we can check between the 30 minutes, 2 hours, 6 hours, 12 hours, and 24 hours after the first exposure on the drugs. We can see there is no changes on the peak, meaning this drug is a no effect drugs. But then when we test sunitinib on the cardiomyocyte, so sunitinib is one of the anti-cancer drugs which inhibit the tyrosine kinase inhibitor. So once we treat with the sunitinib, you can see that the cardiomyocyte is dying, that the peak it become first stronger and then slower, and slower. So you can see the effects of the drugs on the cardiomyocyte based on the prolonged chronic exposure. So this is one of the projects that I have been involved. It's called the Crack It Impulse Challenge. So the Crack It Impulse Challenge has been coordinated by my supervisor, Professor Chris Denning, and it was funded by the GSK, GlaxoSmithKline, and we have been awarded around 1.1 million for the duration of 3 years to conduct this project. So the project is under monitoring of the National Center of Replacement, Refinement, Reduction of Animal in the Research. And together with the technology provider, Clyde Bioscience, who provide the cell optic platform, and Cardia, which is a spin-out company, which doing a commercial cardiomyocyte, which is named as a Pluripotent, together with our academic partner. So the university that involved is the Hamburg University, which has been led by the Professor Thomas Ischenegen, the University of Leiden University, which is led by the Christine Memory, and also Glasgow University, which is led by Professor Godfrey Smith. So this is a picture with me and Christine Memory in one of our meeting about the projects. So this project has been successfully conducted and published last year. You can refer to the paper. So how does this project been done? So it was done in the 2015. We have done a meeting to shortlist the compound for the blinded screen, and then the GSK, the GlaxoSmithKline, has finalized the compound for the compound and prepare them in the brown bottle with the barcoded. So we doesn't know like what drugs arrive to us. Next, the drug has been shipped to all the CRACID partners together with the in-house cardiomyocyte differentiation. So in Nottingham, we have supplied our REPRODUCTIVE Nottingham tailor-made reprogramming cells, and then we differentiate it into cardiomyocyte, and we have sent it to our partners. We have conducted the test into 3 different platforms, into the cell optic platform, which is the 2D, the 3D engineered heart tissue by the Hamburg University, and we have done also in the 2D again with the triple transient measurement by the Leiden University. So in May 2018, we have presented the result from all our partners, and the classification and unblinding revelation was done by GSK to make sure like whether our prediction is correct or not. For example, whether the drug is on the positive anatrophy or negative anatrophy, or it has no effects at all. So what I want to conclude about the cardiomyocyte, first of all, to make a cardiomyocyte, you can use it from both pluripotent stem cell, whether it's from the embryonic pluripotent stem cell or human-induced pluripotent stem cell, which has undergone the programming. And if you want to create a disease model, or you want to create a mutation or polymorphisms, you can genome edit the cardiomyocyte to make sure that they are on isogenic. So you kind of can compare like apple to apple. You don't want to compare apple to the orange. So you can do genome editing, and next you can do the cardiac differentiation to make sure it become a cardiomyocyte, a breathing cardiomyocyte, and then you can do the phenotyping, the molecular and the functional phenotyping, especially that related to the heart, and which eventually can be developed more for the drug development or biomarker or diagnostic for the rare disease, including for the mutation, for example, like hypertrophy. So I think that is all for my sharing.
Thank you very much, Dr. Nurul Ain Nasim, for a very nice presentation. I guess you have gone through the description regarding some manipulation that we can do on the pluripotent stem cells after we differentiated them into cardiomyocytes. And this includes the genome edited technology, genome editing technology, and for various purposes, this can also lead to the drug discovery, and also you can apply it for testing some natural products. I think that's the end of the session for all of our speakers. We've done with that. Now let's have quick a Q&A session. I can only allow 1 minute for each presenter to answer the question. So the first question goes to Prof. NORSHARIZA. Are you still there?
Yes, I am.
I think this is Dr. Azim from USM asking, is it crucial to generate the same size embryo bodies, more or less the same diameter? Does it affect the differentiation later on? What is your experience on this? Thank you.
Sorry. Yes, definitely it will affect. The reason being because the different size of embryo bodies will indicate their differentiation potential. For example, for neurogenic potential, the best size is between 300 to 350. So when you when you do the embryo bodies formation, you have to make sure your recipe is consistent, and then you know that the exact size of your embryo bodies, and also that will be determined by the time, because you have to remember the embryo bodies is mimicking your embryo. So embryo development also involve different stages. So you start with ectoderm and then go on to mesoderm, and then end up with endoderm. So endoderm will be the last stage. So if you create your embryonic body size above 400 to 500 micrometer in size of embryonic bodies, they will then have a tendency to differentiate into endodermal lineage. So for us, the ectoderm or the neural lineage is about 300 to 350. You can get you can get all the lineages with different size of it, but the efficiency will be compromised. So it's important. Yeah, my answer is important. It does affect the differentiation later. Thank you for the question.
One more question for you. We are using a different transcription factors for the reprogramming, and how this can affect the pluripotency of the pluripotent stem cells.
Yes. Okay. So if I understand the question correctly, so you mean a different transcription factors for reprogramming, right? So you are
Yes.
not yes. So there are group of people who are now because the original or no the earlier stage proposed by Yamanaka was to use until now most people are using the OSKM, the for SOX to TLF4 and also the CIMIC as the transcription factors for reprogramming. As I mentioned in my presentation just now, so the transcription factors involved or required for maintaining the prepotency, it has different functioning roles, right? Some compensate others, some actually have redundant effects. So that's why if you look at the OKSM, it does not include NANOC, even though NANOC is one of the core prepotency markers, because it actually compensates by being CIMIC and KRF4. Similarly, other extended regulatory factors also actually compensate the need of the core pluripotency markers. So people now have replaced CIMIC with LIN28, and also with other transcription factors, just to make sure that, because now people believe CIMIC, even though it's important, is involved in diverse developmental processes, but it also can stimulate and trigger oncogene expression. So it's actually can trigger what we call teratogenic formation, teratoma formation, and it can transform the cell to cancerous lines. So people have changed it to other factors as well. So and then and also people have used direct inhibition about play around with epigenetic modification, and also people use small molecules. So all these actually it depends on what you are targeting. How are you going to rectify the process, the effect of these processes. So back to the questions whether it will compromise the efficiency. Yes, that's why it's important, because you have to remember the original source. The original source that you use to reprogram also plays an important role. So you need to know your cells, your somatic cells, or your cells that you want to reprogram, because nowadays people also use low potency stem cells to reprogram. So they use, for example, mesenchymal stem cells. Also now people have been used to reprogram into induced pluripotent stem cells, and also the somatic cells. Somatic cells also at a different level of ability and also properties. For example, bone marrow compared to fibroblasts, they have a different properties, and of course, different cells, different somatic cells, they will express different set of genes. Among them, they will include also partly those genes that are required to become embryonic state. So back to the question. So if you're asking if different structural factors will lead to different efficiency, yes. So you need to know the need and also the properties of the cell that you are reprogramming. Yeah. So I hope that answers your question. Right.
Wonderful. Thank you very much, Prof. NORSHARIZA. Now, a question for Dr. Muhammad Lokman. What are the challenges when you're dealing with the endangered species, especially when you want to acquire the sample?
Okay, thank you very much for the question. Yeah, because nowadays we are talking with the SDG, the university. We're talking about Sustainable Development Goal. We have 17, right? But IIUM will add on another one, which is the spirituality. Gonna be 18, yeah. Okay, because of we after we check, and then we mark that there is no spirituality there. So previously we are having the human without soul, product without soul in industrial revolution of 3, and now we are into industrial revolution 4. Yeah, so we try to find out the soul of our products of university and so on. Yeah, and then yeah, when talking about SDG, we're talking about endangered species, we're talking about something that maybe we can lose it, and then we're gonna give nothing to our future. Okay, or robotic, or something else like that. Okay, so that's why I'm eager to have this kind of research, like to have collecting all those kind of piece of the keys endangered species, like everyday life we having, like normally they will call me when like not panda, a tapir being having the vehicle motor accidents, for example, like maybe from the tiger. We have the Malayan tiger is less than 80 in Malaysia, and then maybe by 2025 our gore will be like no longer in the wildlife. Okay, so this is the thing that we have to ensure. Okay, so yeah, back to the question, the most important, the most problem or the crucial part is to grow the cell from the tissue. Okay, because every species, previously we're talking about human, each of human have a different polymorphism and so on, but now we're talking about different species of animal. Okay, every different species of animal will behave differently, so we have to play around with the media, the condition media, to derive the cell. The tiger will have a different types of media condition, concentration, and what so on. And the tapir, the gore, the seladang, or the whatever. And then we are towards the fish as well, the fish and the birds. Okay, not just a mammal. So this is the hurdles that we are facing, but yeah, it's quite exciting. And then I like to do this kind of research, actually. Yeah, so I think that's all about that. Yeah.
Thank you very much. Thank you very much, Dr. Muhammad Lokman, for that lovely answer. Let's move on to the third speaker. There's one question. How similar the native human cardiomyocyte to the one that you have created? Is there any a variation and how you address this issue?
So there are a variation of that, especially with the presence of the T tubule. And then you can see from the maturation, like for example, our heart, we mostly use the fatty acids to metabolize it, not the glucose. So our cardiomyocyte use glucose to metabolize it. So it's mentioned, so it's mean like our cardiomyocyte is not that mature enough compared to the normal heart. And we're still lacking of a few structure, for example, the T tubule. So for example, in my experiment, which I studied the beta adrenergic system, T tubule is really important because the position of the beta adrenergic is only in the T tubule sites. So it's not 100% similar with the human, but at least but now there are a few methods to make it more mature. For example, by using it co-culture with epithelial cell, co-culture with the fibroblast, because it was believed that this fibroblast and fibroblast or epithelial cell has created a secretome, and to make it to support cardiomyocyte, because in our heart it's not cardiomyocyte alone, it has a population of cells. Yeah, so it's not similar with the adults, but we are now trying to make it more or less similar with the adults. Yeah. It still have flaws. It's not a perfect system. Yeah.
Well, we got another question for Dr. Ain. Is it possible if we use either cell optic, TTM, or cardio ECR to detect loss of sensation or muscle function?
I think for the cell optic, if you use for the contractility, yes, we can detect for the muscle function, because I bet that your muscle maybe is twitching, so you can see some kind of because with the contractility is not based on the voltage dial or anything, it's just based on the pixel displacement. It's like you take 1,000 frame of images in 1 second, and then you try to compare whether each of the slide is moved or not. So if you have a muscle function, a twitching muscle, I think you can observe it in the cell optic and in the TTM, but not for the cardio ECR. I think not for the cardio ECR. Yeah. But I think the challenge now is we don't have any of the platform in Malaysia yet. That is the biggest hurdle. Yeah, that's really biggest hurdle. Yeah.
Yes, that's true. That's the main challenges for stem cell scientists, especially, to bring these forwards. And that brings the theme of this webinar. We want to leverage these pluripotent stem cells so that some people in Malaysia, especially, can benefit from it. I think that's it for today. Thank you, everyone. Perhaps there might be more questions from the participants, but I am more likely to advise them to contact you personally, if that's okay. All right. So I would like to extend our heartiest appreciation to all of our speakers. The first speaker, Associate Professor Dr. NORSHARIZA binti NORDIN from UPM. Second speaker, Associate Professor Dr. Muhammad LOKMAN from IIUM. The third speaker, Dr. NURUL NASIM binti Muhammad YUSOF from IMR. Thank you for your participation. And as for the attendees, please feel free to fill out the feedback forms. I know we have a few technical issues today, and also some we are running, or we are behind the time actually, but that's fine. We have achieved our aim for this webinar. Congratulations, everyone. Thank you for your participation. And finally, for the recording, you may log into if you have the Facebook account, go to the IIUM Molecular and Cellular Biology Research Cluster. And for the speakers, we may provide the link personally. For all the registered participants, we will provide the e-certificate after the session. Thank you, everyone, for your attendance and your lovely comments and also questions. Thank you for your time, and hope this Ramadan will fill you with Allah's blessing. Okay, thank you very much. I would like to end the session by recitation of Tasbih Kafarah and Suratul Asr. Assalamualaikum warahmatullahi wabarakatuh. Thank you, everyone.
Thank you.